Users love Correlation Engine for the tremendous amount of expert-curated, omic-scale data that it lays out in a biologist-friendly user interface. Multi-omics data that is continuously added to the system and can be combined with your private data. Translational researchers use the push-button applications to extend their line of sight by generating and validating novel hypotheses. In this training, I'll demonstrate 3 common ways to extract insight. I'll start with a disease search to discover top multiomic biomarkers, then do a gene search to expose new data-driven horizons, and end with showing an example of a pre-processed RNA-seq differential expression experiment and query it to highlight meta-analysis workflows. You will see how to integrate observations across independent studies that add power and magnify biomarker relevance to impact your scientific direction. When you log into Correlation Engine, you land on the home page. In the left panel, users import data, access it in My Data, and share it with collaborators. The dashboard apps in the top panel launch applications. These are where you interact with results after queries are made. Think of Correlation Engine as a deep and interactive life science search engine. Query a gene, a disease, a drug, or results from your team's omic experiments. The central panel is where the action happens. Let's start with a disease search in the Genetic Markers app for psoriasis. As I begin to type, you see autocomplete drop-down appears to help identify standardized concept terms of interest. The terms and types are based on underlying ontologies and dictionaries. Free text search is also an option. Here is a list of the top data-driven markers for psoriasis using the Genetic Markers app. It pulls together all of the available psoriasis omics data from curated studies and ranks biomarkers using a multi-omic machine learning algorithm. You'll hear me say rank frequently. The rank-based methods will become clearer. Essentially, they bring the highest significant results to the top. We see LCE3D is the top gene. There are 42 correlated studies where LCE3D is observed to be highly significant. RNA expression is upregulated, and there are genotyping studies to help identify variants. When you click on a gene, a new tab opens with the data that drove it into the list. I clicked on LCE3D, and now there's a new tab, Studies for LCED3. The top study is skin biopsies, lesional and non-lesional, from patients with psoriasis or psoriatic arthritis. As we scroll down, you see all psoriasis studies where LCE3D was significant. We can see 3 levels of information for the correlated and scored results. Level 1 is the study name. It's easy to understand what was investigated. Level 2 shows experimental bioset names and statistics for LCE3D significance in the comparisons. We see 3 experiments where LCE3D RNA is significantly upregulated. The 3 biosets have disease versus normal sample type comparisons. The top row for skin biopsies of patients with lesional psoriasis versus healthy without psoriasis shows differential expression of LCE3D RNA was observed to be upregulated 46.5-fold with a highly significant p-value of 3.4 to the -45th. Within the bioset containing all of the significant differentially expressed genes, LCE3D, based on its rank, received a normalized score of 100. In the second bioset within this study, LCE3D scored 97, and in the last it scored 96. Level 3 is displayed when a bioset is clicked on. Here we see more measures, including the Benjamini-Hochberg adjusted Q value, test expression and control expression values. Most important for the scoring is the gene's rank within the bioset. The top rank of 1 is given to the gene having the highest absolute fold change. The maximum gene score of 100 is given to the top ranked gene, and gene scores decrease linearly as rank descends the list. This normalization step ensures cross-study comparisons at the gene level are on equal footing. The second highest study is skin biopsies from psoriasis patients treated with adalimumab or methotrexate. This one is particularly interesting as level 2 results show that at baseline, before treatment, the 6th and 7th rows, LCE3D RNA is upregulated in lesional versus non-lesional skin. Adalimumab responders show LCE3D RNA goes from upregulated 9-fold at baseline to progressively downregulated, down 2.3-fold at 2 weeks, to down 4.8-fold at 4 weeks, to down 5.3-fold at 16 weeks. Non-responders maintained elevated LCE3D 3.5-fold compared to responders. Correlation engine quickly points out markers that define drug response. The quick view icon will take you to the full list of significantly changing genes in the bioset, where it can be used as a data profile, querying to find other highly ranked signatures and independently confirm biological activities. Going back to the correlated gene tabs and scrolling down, we see other top psoriasis genes and supporting molecular data types. A role for IL-23A in psoriasis is supported by evidence of RNA upregulation, decreases in DNA methylation, and the presence of single nucleotide polymorphisms. A machine learning algorithm determines a marker's significance through a meta-analysis that considers the marker's rank across all studies tagged with the queried phenotype. As such, the multiomic derived lists from the Genetic Markers app produce data-driven multi-gene biomarkers for diseases of all kinds. As translational researchers, we always innovate and are interested in knowing more about gene activities and therapeutic intervention. The LCE3D gene is understudied in the research literature. Let's see what Correlation Engine reveals. Hovering over LCE3D, we see it's a component of an epidermal differentiation complex. This seems to make biological sense for a psoriasis activation gene, where uncontrolled epidermal growth is the major phenotypic hallmark. From here, we can focus on LCE3D by clicking on the Quick View icon. The Quick View app shows the top 5 results from the dashboard apps and our entry points to getting into the data. In the Body Atlas, you will see normal tissue, normal cell types, stem cell, and cell line rankings. For gene queries, the Disease Atlas ranks thousands of diseases by relevance. The most correlated LCE3D related diseases include psoriasis, rhinitis, and other immune-stimulated disorders. The pharmacoatlas shows LCE3D activity is affected by Amgen's anti-IL-17 receptor treatment, brodalumab. AbbVie's anti-TNF adalimumab treatment is scored. The knockdown atlas shows genetically perturbed genes that affect LCE3D expression. This app is particularly useful when investigating upstream regulators and drug on and off target effects. If a gene is knocked out, then after drug treatment, what genes are responsive or remain active compared to controls? The literature app shows just 16 PubMed articles for LCE3D. Our research finds that gene searches in PubMed very often produce low numbers of publications. In fact, thousands of genes produce no hits in PubMed, where Correlation Engine finds thousands of hits exposed in real-world omics data. The tag cloud interactivity is particularly useful in reviewing co-cited terms and articles you want to read. Let's click on the phenotype filter. Keying in on the large text terms, we see LCE3D is co-cited relatively higher in psoriasis and related skin disorders, as well as some cancers. Clicking on a term brings you to those articles. When I click on psoriasis, I see 8 PubMed articles and links to them. The literature app is updated every day. Now let's go to the real-world experimental data in the Curated Studies app. We find 1,655 studies where LCE3D was significant. By exporting results, you'd see 4,500 experimental bioset comparisons where LCE3D was significant. That's about 300 times more hits on data than articles. We can filter by organisms or take a look at data types. We will find there's 896 RNA expression studies, alongside studies from 7 other omics data types where LCE3D was observed to be significant. Here you see the multi-omic potential to better understand biology. For instance, might LCE3D RNA upregulation be driven by increases in DNA copy number, or DNA hypomethylation, or other epigenetic changes? Correlation Engine enables you to discover deeper levels of biology driving changes in gene activities. The top LCE3D study, lesional and non-lesional skin from psoriasis, atopic dermatitis, or contact dermatitis patients, we see a multifaceted experiment, RNA expression experiment with human samples. LCE3D scores higher in atopic dermatitis, disease versus normal comparisons, than psoriasis and contact dermatitis. It is upregulated in all 3 phenotypes. Scoring normalizes for omics platform differences across studies and for variable numbers of significant genes after removing insignificant genes. Links to our original 2010 article, Ontology-Based Meta-Analysis of Global Collections of High-Throughput Public Data, and other resources will be shared at the end of this tutorial. The second study is skin tissues of patients with mild extrinsic or intrinsic atopic dermatitis or mild plaque psoriasis. It compares mild forms of atopic dermatitis and psoriasis to skin samples from healthy, non-psoriasis subjects. LCE3D RNA is again observed to be upregulated in these three comparisons. The third highest ranking study, skin biopsies of plaque psoriasis patients treated with brodalumab or ustekinumab, compares a wide range of psoriasis disease versus normal and biologic treated versus control experiments. In total, 47 bioset comparisons were made from the available sample types. In a nutshell, LCE3D RNA was upregulated in baseline disease versus normal biosets. And over a time course with brodalumab or ustekinumab, LCE3D RNA is observed to reverse from upregulated at baseline to downregulated. Over a 12-week administration of brodalumab or ustekinumab treatments, LCE3D RNA levels progressively decrease at 4 and 12 weeks. Given these changes, the drug targets for IL-17, IL-12, and IL-23 appear to be upstream regulators of LCE3D gene expression. As interleukins are widely pleiotropic, often implicated in hundreds of diseases, might targeting LCE3D more specifically treat psoriasis? The fourth ranking study, skin biopsies, lesional and non-lesional, from patients with psoriatic or psoriatic arthritis, is another independent series of RNA expression experiments comparing psoriasis to normal human skin. And here we see 4 comparisons. LCE3D RNA is upregulated in both psoriasis and psoriatic arthritis, except for no measured significance in lesional psoriatic arthritis versus unmatched healthy without psoriasis. By clicking on full study details, let's look more deeply into this curated study and the available comparisons. Our curation team does a rigorous analysis on publicly available studies, like this one from Gene Expression Omnibus. It was run on an Illumina NovaSeq 6000. Curators give studies an easy-to-understand name, carry over and annotate source descriptions, compute test versus control comparisons, document quality control metrics, and post signatures based on the available sample varieties within the study. This one included lesional and non-lesional skin samples from 13 patients with psoriasis and 15 with psoriatic arthritis. Healthy skin from 12 patients also served as controls. At the bottom, we see curators' notes. Note 1 lists comparisons that did not produce significant results and were not included. Note 2 further documents our rigor. 2 mislabeled samples were detected in the public repository. The authors were contacted for clarification, but did not respond. As there were sufficient replicates, those samples were removed from the analysis. Let's open the Biosets tab. Biosets are what we call the end results from sample comparisons and the associated metadata. Here we see 4 biosets clearly named so the experiments are readily understood. Files associated with quality control and the analysis are also available here and under the Images tab. I'll click on the first bioset. For skin biopsies of patients, lesional psoriasis, versus healthy without psoriasis to present a better picture of what a bioset provides. By clicking on the View Bioset Details, we see how the data was processed and the cutoffs used to remove insignificant genes. For uniformity, curators adhere to omics data type and platform-specific processing method. A list of significant genes and metrics are featured here. For RNA expression data, genes are ranked by highest absolute expression. 6,309 genes passed significance cutoffs. LCE3D is ranked number 2, with a positive 46.5-fold change between disease and normal samples, with highly significant P and Q values. The test expression and control expression values provide the RNA abundance levels used to calculate fold changes. Every bioset is tagged with the essential experimental concepts. We see the design is disease versus normal. Samples came from primary tissue, fresh or fresh frozen. The phenotype is psoriasis, and the tissue is skin. Tags are used throughout the system's search, filtering, and machine learning results, as we saw in the genetic markers app, where we started this tutorial. It's a good time to point out that you can export data throughout the system. A complete set of application programming interfaces enable data scientists to query, retrieve, and integrate correlation engine results into their engineered work streams. Now, instead of querying a disease or gene, let's correlate this bioset to every other bioset in the system by going to the Curated Studies app in the dashboard. In essence, we're searching data with data. We see over 23,000 curated studies ranked by highest bioset correlation significance. Under the data types filter, we see the distribution of curated studies by molecular modality. There are over 13,000 RNA expression studies that cover various sequencing and array platforms across a wide breadth of biology. We see over 1,600 genotyping SNP-GWAS studies. There are DNA copy number, DNA methylation, histone modification, protein DNA binding, and ATAC-seq, microRNA, and correlations to other data types. Multiomic correlation is inherent in the works. To make better decisions, publish, and write better grants, investigators need a simple means to extend their reach. Correlation engine does that. For instance, if one has generated and imported RNA-seq differential expression data, they can combine it with upstream somatic mutation, DNA methylation, DNA copy number, histone modification data, and downstream with proteomics data. As we scroll down, of the 7,000 different phenotypes tagged to data in Correlation Engine, we see top correlated studies are psoriasis studies. These are data-driven results. We did not filter for them. It's the similar psoriasis biology captured in the experimental omics data that drove them to the top. The top scoring bioset correlation when querying this lesional psoriasis skin ranked list of 6,300 significant genes was to another bioset in the same study. The lesional psoriatic arthritis skin signature correlated higher than any of the 190,000 biosets in the system. So it was given a normalized score of 100, while other biosets are scored by significance in descending order. When I click on a bioset name, Level 3 results appear. A summary of the gene counts and expression direction is shown for the queried bioset, bioset 1, and the discovery bioset, bioset 2. The Venn diagram shows a large overlap of common genes and some outliers that may be disease-specific. The bar chart breaks down the directional results. Correlation Engine uses a novel running Fisher algorithm that uses gene ranks to calculate a p-value for the positively correlated genes that are either up in both or down in both. Negatively correlated genes have opposite expression directionality. No negatively correlated genes were observed in this bioset comparison. The table below the graphs show the rankings between the biosets. This table is exportable, as is export of a table with all 23,000 level 1 and 116,000 level 2 results with metrics. Interestingly, LCE3D was insignificant in the psoriatic arthritis biosets. The ontology-based results in the disease atlas ranks disease phenotypes driven by disease tags and a disease versus normal experimental design tag. We see that psoriasis is at the top, recalling that we saw top curated studies were from other psoriasis studies driven by the strength of all bioset correlations in the system. The default view lists top 5 diseases in disease categories. You see that psoriasis is both a skin disorder and an autoimmune disease. By clicking on View by Ranks, we see the high-level results for 614 diseases. Beneath psoriasis, the queried lesional psoriasis versus healthy without psoriasis bioset has large overlap with biosets from other skin disorders and infectious diseases. These phenotypes have large innate and adaptive immune response components. Correlation Engine provides high-value tools and insight to explore similarities and differences. We see 71 psoriasis studies correlated across independent RNA expression, DNA methylation, and SNP-GWAS experiments. There is even a pharmacoatlas that pulls together and ranks results from over 6,000 distinct compounds tagged in treated versus control experiments in the system. Here, therapeutic response measured across omics data types can be readily assessed for mechanistic insight. Let's end this training with what I consider to be the most profound application: aggregating biosets for adding power beyond a single experiment using the meta-analysis app. Users validate their imported data by comparing it to similar experimental designs in the expert-curated public data. While still in the disease atlas, we'll click on psoriasis. The meta-analysis app is engaged by clicking on the blue funnel icon. I'll click on the icon next to the queried bioset. Then you see a 1 in the dashboard app. The first study, as we've seen previously, is from correlations to another bioset in the same study. So let's skip that one. Now I'll quickly go into the next 4 studies and click on the meta-analysis icon next to the bioset names. In skin lesional and non-lesional from plaque psoriasis patients and skin from healthy subjects, there's a bioset, skin lesional plaque from psoriasis patients versus skin from healthy control subjects. I'll click the blue icon to add that. The next study, skin biopsies from patients with psoriasis. Again, another disease versus normal comparison in human RNA. And then skin tissues of patients with mild extrinsic or intrinsic atopic dermatitis or mild plaque psoriasis. There's a mild plaque psoriasis bioset. Let's click on that one as well. Lastly, skin lesion and non-lesion areas of psoriatic patients treated with AHR agonist and antagonist. There's a disease versus normal bioset in there as well. Let's click on that blue icon. Now back to the dashboard app, we see in number 5 in the meta-analysis, our queried bioset plus 4 other biosets. The default tab is for setup. Here we can move bioset order up or down, or select for up or down regulated genes, only up regulated genes, only down regulated genes, or genes that are absent from the biosets. Opening the Gene Results tab, we see in the micrographs that top psoriasis markers are upregulated across independent experiments. For each gene in rows, there's a bar for the 5 biosets selected. A bit further down, we see that keratin 77, KRT77, is downregulated across each of the biosets. Our gene of interest, LCE3D, is ranked 5th. Opening to level 2, we see LCE3D RNA scores very high in each of the upregulated fold changes. Level 3 results show LCE3D rank within each bioset ranges from 2nd to 50th. The normalized gene score, while quite high, descends accordingly. In conclusion, I hope you've gained a better understanding for how Correlation Engine works. I encourage you to think about your science and imagine what Correlation Engine would reveal toward a better understanding of wide and deep mechanistic biology. Thank you.